Hey guys, my name is Kalu Dike Udensi and I welcome you to my presentation on the construction of an SCO3855 mutant. Streptomyces are bacteria characterized by their developmental cycle, which contains the complexity of a multicellular organism. There's a drastic change in the direction of growth as a response to signals received that results in the upward growth and production of secondary metabolites. The presence of rhomboid proteases, a signaling molecule, suggests the direct involvement in the developmental cycle of streptomyces. Rhomboid proteases are enzymes that catalyze the cleavage of peptide bonds. SCO3855 is one of four protective rhomboid proteases found in Streptomyces cerevisiae, a model organism for streptomyces used by the Trujillo lab in our quest to understand the role of rhomboids. Over here, we observe the proposed model of cleavage mechanisms for rhomboid proteases. Putative SCO3855 was successfully cloned in previous experiments, and with the help of individuals from Brooklyn College, we developed the CRISPR model structure based on the CRISPR model of GLPG, a rhomboid protease grown in E. coli. The highly conserved nature of rhomboid proteases allows for the development of this model based on a similar one. Let me call your attention to the center of both models, which contains on the left-hand side the serine structure and on the right-hand side histidine. Here we can observe the structure of serine and histidine at the active site, serine being the red and histidine being the blue. Based on research done by previous groups, we understand that mutation of serine to threonine makes the enzyme inactive by blocking the access of the substrates to the enzyme. The inactive SCO3855 would be our negative control for the characterization of rhomboid proteases. To achieve the mutation, we target the specific sequence of serine that codes for the amino acid. This experiment is called site-directed mutagenesis. The objective of site-directed mutagenesis is to mutate the serine to threonine. To begin, we designed two primers, one containing the exact sequence that we aim to generate, and the other one containing the new nucleotide that codes for threonine. Using PCR, we amplify the plasmid, then we follow the listed steps. At the end of these steps, we gain two different templates. One is an inactive mutant, cut by the agel, and the other one is a wild type, not cut by the agel. We destroy the wild type, not cut by the agel, and then we transform the inactive mutants into cells for growth and transformation. Over here, we observe the two differences when sequence of the SCO3855 and the inactive SCO3855 after the threonine has been substituted for the serine. We can see the difference in amino acid sequence in the first one compared to the second one. The next step is to perform an enzymatic assay to compare the positive control with the negative control when it reacts with a substrate. When the positive control, which is a putative SCO3855, reacts with a substrate, there is a cleaved PS-TAT-A, and when observed, there's a change in the emission spectrum produced. However, when the inactive SCO3855 reacts with a substrate, there is no cleavage in the PS-TAT-A, and there also is no change in the emission spectrum. This newfound knowledge on rhomboid protease that I was able to gain and also share with you guys was made possible by these few individuals and corporations: QCC, NSF, CUNY Research Program, C-STEP, Dr. Monica Trujillo, and the former students for their experimental work done, Max Steinhartman and Janine Seng from Brooklyn College, UCSF for their program used in designing a molecular model, and of course, CRIPS for the funding and opportunity to perform and present my research. Thank you.